seventh Nobel Symposium organized by CSIR-CDRI. As it is globally known since 1901, Nobel Prize has been presented to the laureates on 10th December every year in the fields of chemistry, zoology, medicine, peace and economics. Considered one of the world's most prestigious awards, it is awarded as per the will of the Swedish-born inventor, chemist and industrialist Alfred Bernhard Nobel. Similarly, Nobel Symposium held in CDRI is one of its kind. It was initiated by Nobel Symposium held in CSIR-CDRI is one of its kind, which was initiated by the former director, Professor Stephen in the year 18 to motivate and inspire the young researchers of CDRI, especially in the fields of chemistry and nutrition. I request our director CSIR-CDRI to come on the stage. Mr. Sarpon and Ms. Swan are requested to present to the Deputy Director. I request Director Ma'am to please accept the token of appreciation. Now, I would like to invite respected Ma'am to formally inaugurate this symposium with her inspiring words. Thank you, ma'am. We are grateful for your kind words and your unwavering support and guidance have always been instrumental to shape the institution. Now, let's begin the session with the Nobel Prize in Chemistry 2004. This year's Nobel Academy of Sciences has decided to award four. This year's Nobel Prize is really exciting as it shows how chemistry is widening its wings by incorporating other disciplines. This year's Nobel Prize is really exciting as it shows how chemistry is widening its wings by incorporating new disciplines like computer science to explore newer technologies are being applied to the fields of basic chemistry to get a better understanding of biological molecules. Thus, we see modern science is no longer recognized by the narrow boundaries. These kinds of interdisciplinary science will lead to groundbreaking research. Yes, sir. Thank you, sir. So, a very good afternoon to one and all. I, Shilpa, working under the supervision of Dr. Richa Pandey. Working under the supervision of Dr. Richa Pandey, will be moderating the session 1. So, today I welcome you all to the 7th Nobel Symposium organized by CSIR-CDRI. As it is globally known, since 1901, Nobel Prize has been presented to the laureates in the fields of chemistry, physics, physiology, peace, literature and economics on 10th December. Considered one of the world's most prestigious awards, it is awarded as per the will of a Swedish-born chemist, inventor and industrialist, Alfred Bernhard Nobel. Similarly, Nobel Symposium organized in the CSIR-CDRI is one of its kind. It was initiated by our former director, Professor Tapaski Kundu, in the year 2018 to motivate and inspire the young researchers of CSIR-CDRI, especially in the fields of chemistry and medicine. Now, I request our director, CSIR-CDRI, Dr. Radha Rangarajan, to come up on the stage. Mr. Sarvabhaum and Swamim are requested to present a bouquet. May I request ma'am to please accept the token of appreciation. Now, I would like to invite respected ma'am to formally inaugurate the symposium with her inspiring words.
Thank you, Shilpa. Good afternoon, everyone, faculty, staff, and my dear students. Students, this is actually your day. You put this symposium together every year with great enthusiasm. In fact, I heard that this year, and perhaps this is true for past years as well, this symposium is highly oversubscribed, meaning that there are many more students who want to participate than can actually make it. So it's on a first come, first served basis. So you're rushing to get your name in line. This is very heartening to hear, and therefore taking this as a positive sign, I will announce some initiative for 2025 that builds on this energy and enthusiasm. Well, today we're going to hear about the scientific journeys that led to Nobel Prizes in physiology or medicine and in chemistry. Can you hear me? We of course salute the courage, creativity and perseverance of those who have won the prizes, remembering that there are others also who participated, and science is a very large team effort. Nevertheless, the prize remains one of the most coveted, most prestigious prizes in basic sciences. I don't know how many of you know about the history of the Nobel Prize. I'll just take you through a very quick history lesson. Alfred Nobel was born on October 21st, 1833 in Stockholm in Sweden. He had a curious mind and had an interest in solving tough problems. As he grew up, and he lived in in Russia, he lived in Sweden, he lived later in France. Anyway, he had a deep interest in chemistry and he was fascinated by nitroglycerin. Now, nitroglycerin is a very explosive material, and it is not easy to handle or to use. He decided to solve the problem of how to control nitroglycerin to enable controlled blasts. And therefore, he started to work on the chemistry of nitroglycerin, and by a systematic process of step optimization, he was able to develop a technique. He filed a patent in 1866. He then went on to found factories, many of them, producing nitroglycerin and several other products that went on to have enormous usage in mining, construction work, tunnel work, etc. But I'm sure you can all recognize that this technology could also be used for arms and ammunition. And he himself also set up factories for arms and ammunition, although he claimed to be a pacifist. And his thinking was that owning arms and ammunition, or countries having stockpiles of these, would be a deterrent for other countries to attack. Now, we know how that goes. So as his fame and wealth grew, and his nitroglycerin-based explosives made their way into many industries with many applications, they also made their way into wars. People were killed, and so on. A turning point for him came in 1888. It turns out his brother Ludwig died in France, but the French newspaper that reported it thought that Alfred Nobel had died, and therefore they wrote an obituary for Alfred Nobel. And they criticized him heavily in this obituary, calling him a merchant of death. So the obituary condemned him for his inventions, criticizing him as the wealthiest vagabond in Europe who had become rich by finding ways to mutilate and kill innocents. Of course, this error was later corrected, but life gave him this rare opportunity to read his own obituary at that point in time. And he was horrified by it. It left a huge mark on his conscience, and he decided that he needed to rectify his legacy. He did not want to be known throughout the world for being a facilitator for horror and death. And so all that he had accumulated began to feel like a burden for him, and he began asking himself, what is the legacy I will leave behind? So he then vowed to work for humanity and decided that he would reward all those who have selflessly worked for society and have conferred great benefit to mankind. Therefore, he rewrote his will, setting up the Nobel Foundation and officially donating almost all of his money, which at that time was 35 million Swedish kroner, estimated to be about $255 million in today's time. And the will said that the Nobel Foundation will give the prize in 5 categories, as we just heard: chemistry, literature, medicine or physiology, physics, and peace. And the first set of prizes were awarded in 1901. And in 1968, an additional prize was added, and that was for economics. So this is the history of the Nobel Prize, and this is why this is actually how the world received such a gift. Sometimes it's useful to read about oneself passing away ahead of time, perhaps. So today we'll hear about the scientific journeys of the Nobel laureates David Baker, Demis Hassabis, John Jumper, Victor Ambros, and Gary Ruvkin. We will learn about protein folding and the role of AI and ML in predicting protein structures. We'll also learn about microRNAs and how they modulate gene expression. The students who are participating today: Abdul, Ashrabuja, Imran, Neha, Pranoy, Shilpa, and Dipanshi. We are very excited to hear from you. And I will not speak for much longer, but before handing it back to the students, I want to acknowledge the role of the mentors: Dr. Pratap, Dr. Imran Siddiqui, Dr. Kinshuk Srivastava, and Dr. Amir Nazir. Your efforts are greatly appreciated. So students, it's over to you. We look forward to an exciting journey today. Thank you.
Thank you, ma'am, for your kind words. We are truly grateful to your kind words, and your unwavering support and guidance has always been instrumental in shaping our institution. So let's begin with the session 1, that is Nobel Prize in Chemistry 2024. So this year, the Royal Swedish Academy of Sciences has decided to award prize with one half to an American scientist, David Baker, University of Washington, Seattle, for computational protein design, and the other half jointly to Demis Hassabis and John M. Jumper, both at the Google DeepMind, London, UK, for protein structure prediction. So this year's This year's Nobel Prize is really exciting as it shows how chemistry has widened its wings by incorporating the other disciplines like the computer sciences to its fold. Newer technologies are being applied to the basic chemistry to give a better understanding of the biological molecules. So, thus we see that the modern science is no longer restricted to the narrow boundaries. These kinds of interdisciplinary science will lead to groundbreaking research and findings. So, let's begin. To begin with the session, let's listen how the Nobel laureates have reacted for the first time when they hear their names for the Nobel Prize.
Hello.
Hi, it's Adam Smith again from the website of the Nobel Prize. Many congratulations.
Thank you.
Can you just tell me how the news reached you?
Let's see. Well, actually, I think it's kind of funny. I didn't realize this till afterwards, but I think they had the phone number for my son, who then gave them my phone number, and so I got a phone call, and my wife promptly started screaming, so I had a little hard time hearing, but then they got the news across.
I like that she twigged before you did.
Yeah.
Hello, Demis. This is Adam Smith from the website of the Nobel Prize.
Great to hear from you.
Many congratulations.
Thank you so much.
You have had so many achievements and accolades, and you're so young, but what does this prize mean to you?
Well, I mean, it's unbelievably special, and you know, it's actually really surreal, to be honest. It hasn't really sunk in, but yeah, and it's an incredible honor. It's the big one, really.
Hello, am I speaking with John Jumper?
Yes.
This is Adam Smith calling from the website of the Nobel Prize. Many congratulations.
Oh, thank you.
I detect in your voice some surprise. Can I just ask you how you feel about this?
Oh, it's absolutely extraordinary. I think it's a wonderful group. I think it's like, I've been a computational biologist a long time, and you know, I like to say in talks, we need this to work. You know, we need computation to solve the problems of biology.
So, let's start with our first speaker, who is Abdul Basit Khan, who is SRF working under the supervision of Dr. J.V. Pratap. Today, he will throw in the light on why researcher community was struggling with the problem of protein puzzle over the half century after Christian B. and Finson's discovery, which demonstrated that the 3D structure of a protein is encoded in the sequence of amino acids. Welcome, Mr. Abdul Basit. The floor is now yours.
Thanks, Shilpa. Good afternoon, everyone. I'm Abdul Basit, and I'll be talking about protein folding, cracking nature's origami code. As Shilpa told, that this year the Nobel Prize in Chemistry 2024 has been conferred to the recent advancement with the help of computation in structural biology, that is protein structure prediction and computational protein design. As she also mentioned, that though this structural biology is based on the core concepts of the biology, it deals with the biomacromolecules, but the prize has been given in the chemistry. And it happened many times. The structural biology technique started from the discovery of the X-rays by Rontgen in 1901, for which he was conferred with the first of the Nobel Prize in Physics. Later on, with the application of the physics, the tools, and the concepts of the chemistry which were later known, like, for example, the covalent and non-covalent bonds, bond length, bond angles, kind of degree of freedom of rotation, it helped the structural biologists to predict, to elucidate the structure of the protein and to find out the folding. Then came the era of the computations. There was a surge in information technology, and it led to the development of a new type of domain in the science that was called as bioinformatics. And later, with the involvement of deep learning and artificial intelligence, it helped us a lot in finding out the structures of the protein in a predicted way and in a quick and efficient way, for which the Nobel Prize this year has been conferred for the discovery of Rosetta and AlphaFold 2. And this made this structural biology and a totally interdisciplinary science. As we were discussing about that structural biology and the Nobel Prize was related to the proteins, let us quickly recapitulate what does the central dogma of the molecular biology suggest. The information encoded in the DNA is transcribed into an mRNA, which is translated into a polypeptide. Then this polypeptide, which consists of number of the amino acid, folds into a tertiary structure of a protein. This folded structure of a protein make it as a life building block, a structural and functional component of the cells, the life processes of all the life domains. That made it, that helps us to understand its role in all the disease pathogenesis. That's why we target proteins for therapeutics. We talked about protein sequence and protein structure. It all started when Christian Enfinson, who was studying the structure of the bovine ribonuclease A, and he found out that the structure is encoded by the sequence of the amino acid in the polypeptide. This helped him to give a postulate that in favorable conditions, a polypeptide sequence is transformed into a tertiary structure of a protein, which is having minimal state of energy as well as it is thermodynamically stable. Parallelly, the research was also going on the function of the ribonuclease A, which was it was done by Moore and Stein, and they found out that the structure of this protein is responsible for the function of the protein. And for this collective, we can say the result which suggested that the sequence defined the structure which defines the function of the protein, they were conferred with the Nobel Prize in Chemistry in 1972. Now we know that there is a polypeptide sequence which is composed of number of the amino acid. It depends on this every type of protein, each and every protein. And this string where the amino acids are attached with the help of the covalent bond called as a peptide bond. And each peptide bond has a kind of degree of freedom of rotation with the help of psi, phi and omega angles, which help this peptide bond to fold. If we ignore the freedom of rotation of these the angles present in the side chains, let us take an example of 100 amino acid chain, which Leventhal has discussed in his paper. He said that if these three conformations are possible with this help of these three angles, there will be 3 raised to the power 300 types of conformation for this particular protein. In simpler terms, you can say it will be 10 raised to the power 143 conformations. That will take 10 raised to the power 136 years for just this polypeptide sequence of 100 amino acid to fold into a tertiary structure, which is much, much greater than the lifespan of whole of the universe. That make it as a time scale challenge. And it is observed that a protein folds in a fraction of seconds, from milliseconds to seconds, we can say. This is a paradox in itself. Leventhal gave solutions for this paradox, but he said that it's an energy landscape diagram in which he suggested that a polypeptide chain, which is an unfolded conformation of a protein, folds into a native structure which is having minimal state of energy, a lesser entropy. That makes it stable. Another thing he suggested, the hierarchical folding, in which there are local folding on the basis of like 25 to 30 amino acids in a protein. They fold together to form local foldings, and these local foldings are clumped together to form a global folding. He tried to address this protein folding problem, but what is this problem? We know that amino acid sequence is folded into a tertiary structure. But the question arises, what exactly is the folding code? What is the folding mechanism? And can we merely on the basis of the sequence, can we predict the structure of a protein just with the help of a sequence? Thanks to Professor G.N. Ramachandran for Ramachandran plot, in which he discussed about the secondary structures of the protein, and he gave an insight about type of conformations possible on the basis of these secondary structures. And if we really are able to find out and we are able to solve this whole of this protein folding problem, then we can go into protein misfolding in neurodegeneration disorders. We can find out the mechanism of protein aggregation in aging, and we can study why does this happen in vitro, that proteins precipitate, but it does not happen in the cellular proteasome, though it is available in the ample amount. Along with that, we can be able to design any of the protein we need as per the therapeutic approaches or any other examples we will be studying or we will be seeing in the later presentation. And we can define or we can determine the structures of the protein. The folding problem was first addressed with the help of the experimental techniques, for example, the X-ray crystallography, nuclear magnetic resonance, and cryo-electron microscopy. Thanks to these techniques and the people behind these techniques, right now we are having 200,000 experimental structures in the protein data bank. With the help of these techniques, we have been able to find out the functional state of the protein. We have been able to study the interaction of the protein with the ligands, for example, the substrate and inhibitors. We have been able to see the mechanism of the proteins, how does this interact with the ligands, with other proteins. We have been able to study the kinetics and dynamics thanks to the recent advancement in the structural biology techniques. But there are some hurdles. It takes a lot of effort and lot of time for a protein's structure to be determined through these experimental methods. These are quite tedious, expensive of course, and the sample preparation is not that easy. There was a surge in computational technology, information technology. It helped structural biologists to develop 3 techniques that are approaches, I would say, homology modeling, threading, fold recognition, which is also called, and the ab initio approach. Out of these 3 techniques, the homology modeling and fold recognition are based on the template modeling, or we can say comparative modeling. In homology modeling, a target sequence is aligned with the homologous protein, which are already present, the structures is present in the protein data bank, and the structure is then predicted on the basis of the homology with those structures. Whereas in case of the fold recognition, unlike the homology modeling, there a whole global protein structure is predicted on the basis of the homology with the global protein. Here, the local folds are analyzed on the basis of their folding, what type of secondary structures they are forming, and they are aligned and they are like kind of overlapped with the structures already present in the protein data bank, and then a structure is predicted. Then comes the ab initio approach. In this one, a sequence of the amino acid is folded into a protein structure. This is predicted by minimizing the energy and playing with the molecular dynamics of the polypeptide chain, which help a structure to be predicted. Here the Leventhal comes again. He is the man who is behind the first ever approach of predicting protein structure. He's the man who brought two of the domains of the intelligence, human and artificial intelligence, to come together in structural biology. He developed an oscilloscope, a kind of model predictor, which he called it as a clutch. And here you can see, this is the crystal structure of the myoglobin, which was elucidated by Kendry and Perutz, for which they were given Nobel Prize in 1962. And he predicted the model of this structure with the help of the oscilloscope clutch he developed. And on this basis, he was able to suggest that, or gave a prophecy that in the coming time, we will be able to find out the structures of the protein with the help of these computational approaches. But these prediction methods we discussed are not that atomic accurate. They're not that efficient as the experimental structures. Then came the era of the deep learning, in which the artificial neural network is developed that makes the machine to learn and to take the decision on its own. If a sequence is given, it can predict the structure on its own without operating it from outside. So it was inducted in the structural biology. We know the computers read only the binomial numerals. It doesn't read the sequence of the protein, doesn't read alanine, valine, glycine, these things. It studies and it digitizes those sequences into binomial numbers, and the physical and chemical constraints that are attached with that amino acid sequence are embedded as word representation, and then altogether they are represented as the dense vectors. And on the basis of these dense vectors, the machine is able to find out the nearest neighbor of a particular amino acid, either attached with the peptide bond covalently or through non-covalent interactions. This leads to a development of a predicted structure. This technique helped us a lot. With this, we have been able to design proteins of interest, and we have been able to predict structures in millions, and in a very quick and efficient way, which are almost near to the atomic accuracy. And this led to the Nobel Prize this year in 2024, which has been conferred for the discovery behind Rosetta and AlphaFold, respectively. And this will be discussed in detail by my fellow colleagues, Ashtbhuj and Imran, who will be discussing about Rosetta and AlphaFold. That's all from me. Thank you.
Thank you, Mr. Abdul Basit, for wonderfully overviewing about how difficult it was to understand the science behind protein folding. So, as described by Mr. Abdul Basit, we have a little information available about the protein folding, which has been a huge roadblock for the protein research. Researchers have also been trying to design a new protein for various applications. So, that is when Professor David Baker has bridged the gap. To get the detailed view of his research journey, I am inviting Mr. Ashtbhuj Mishra, who is working as a SRF with Dr. T. Narendra. Let's look how Professor Baker turned the table in 2003 when he designed a completely new kind of protein which never exists. Let's hear from Mr. Ashtbhuj Mishra.
Thank you, dear Silpa, for introducing me and inviting me over here to speak. Good afternoon, everyone. Today, I'm going to talk about David Baker's journey from folding to de novo design of protein. Dr. Baker was born on October 6, 1962 in Seattle, USA. He did his schooling from Garfield High School, Seattle in 1981, followed by his BA with a major in biology in 1984 from Harvard University. Then he did his PhD from UC Berkeley in 1989, followed by his postdoctoral training in 1993 from UCSF. In the same year, he joined University of Washington School of Medicine, where he is currently serving as professor of biochemistry. In his doctoral research with Dr. Randy Sackman, Dr. David Baker understood the function and transport of protein in yeast extracts and published his findings in Cell and Cell Press. In his postdoctoral research, Dr. David Baker joined Dr. David A. Agard at UCSF, and there he understood the protein crystal structure, the protein crystallography. Once he got the idea of protein structure and function, once he got the idea that function is being derived on the basis of structure, this idea further deepens in his mind that can be design a new protein with a new function, and with this vision he joined the University of Washington, and there he created a plethora of proteins, as you can see in this slide. The barrel-shaped protein, the double helical and triple helical protein, the protein with different symmetry like C3, C4, C5, and many more type of proteins which were never found in nature. And this led him to fetch the Nobel Prize of the year. Before I tell you that what is protein design, let me give you a brief of protein structure and protein design. Proteins folds into its unique native structure and functions in that structure only, as discussed by my colleague Bassett. So a given sequence of polypeptide chain folds into 3D structure, and this folding process undergoes through many energy barriers, and this is why it becomes really a big problem to predict the structure of protein. Another big problem is, for a given structure of protein, for a given fold, can we bring a new function by designing a new sequence? And this is the problem of protein design, or inverse folding problem. But this is not an easy piece of cake. Designing a protein have several challenges. And the first challenge is, for in an amino acid, there are many rotatable bonds. Single bonds are rotatable, and each rotation can give a new conformation. So it creates an astronomical number of polypeptide chain conformations. Another big problem is, there are 20 naturally occurring amino acids, and different permutation and combination of these sequences can create an astronomical number of proteins. The third big challenge is determining the system level energy. Protein contains thousands of molecules which interacts with lots of atoms, and determining energy interaction to attain a global energy minima is another big challenge. And to address these challenges, David Baker came up with two solutions. The first solution is searching through conformations for a fixed sequence, which he did by software, Rosetta software suite, as you can see in this video. In the second solution, he fixed a structure by searching through all the possible conformations. And in third solution, he did rapid energy calculations like Lennard-Jones potential, calculation of solvation energy, hydrogen bonding, electrostatic interaction of negatives and positives, and torsional preferences. With these approaches to solve the challenge of protein design, David Baker developed a software suite called Rosetta, which is being used for protein design and structure prediction. So the very first step in protein design is designing a backbone of interest to do a function of interest. The second step is designing the sequence that can result into the backbone of interest. And the third step is redesigning and optimizing this sequence and backbone together for further improvements. This is the timeline for development of Rosetta. The journey of Rosetta begins in 1994 with the inception of Rosetta. A major breakthrough came in 2006 when Rosetta was started being used for protein design. Before this, Rosetta was being used for structure prediction only. In 2010, Rosetta Home and launch of Foldit occurs, which is an online multiplayer game. And TR Rosetta was launched in 2021, and finally in 2024, Rosetta Fold 2 was launched. So to validate his Rosetta tool of backbone design, David Baker published a paper, JAX, in 2002, where he designed several protein structures using Rosetta, and you can see that the structure design model created by Rosetta is very similar to the native structure. A major breakthrough came in 2003 when David Baker, along with his student Brian Kuhlman and several other students, created a completely new globular protein with atomic level accuracy. This protein consists of 93 residue of amino acids and is given the name of TOP7, topology of protein structure 7, with a novel sequence and topology. The X-ray crystal structure of TOP7 was almost similar to the design model with a very small RMSD of 1.2 angstrom, root mean square deviation. This is a 2D schematic diagram of designed model. This one is a stick diagram of design model and X-ray crystal structure. This is backbone and protein overlay diagram of design model shown in blue and X-ray crystal structure shown in red. And this is C-terminal of a few amino acids, overlay diagram of crystal structure and design model, and in all three you can see that they are, how beautifully they are overlapping on each other with a very small root mean square deviation of 0.79 angstrom in this C-terminal case and 1.2 angstrom in this backbone. So with this, this first noble globular protein opens the doors to much more possibilities in protein design, and the reason for which he got the Nobel Prize is, till that time, till 2003, the other people, the other scientists who were working on designing the protein, they were actually redesigning the already existing natural proteins to enhance their stability and to bring new function. It was the first time that someone came up with a completely new protein, which was at that time was considered as almost impossible thing. So nowadays we are facing the challenges that were never faced by us during natural evolution. To address these challenges, can be designed a complete new world of synthetic proteins to address this. So keeping this vision in mind, Baker Lab is working on designing several type of proteins like mini binders, and designing enzymes for catalysis, designing peptides as inhibitors, designing nanomaterials for better drug delivery, designing hybrid materials for energy. These are several recent findings published in various good articles. Let me focus on one such application where other people used David Baker's Rosetta tool to design a mini protein binder to neutralize a bacterial infection. Clostridium difficile is a bacterium that causes infection in the colon, leading to symptoms ranging from diarrhea to life-threatening problem. C. diff infections typically occur after using antibiotics. In this, there are two key virulent factors which causes all the infections of Clostridium difficile, the TCDB1 and TCDB2. They designed mini binders, first targeting the first virulence factor TCDB1, and they created 69,752 designs, almost 70,000 designs, which were neutralizing the TCDB1 variant, and then they applied these 70,000 designs on TCDB2, and they obtained 3,434 pan-specific designs, which were neutralizing both TCDB1 and TCDB2 together. So till now, large sub-proteins have been designed, but the possibility of proteins that can be designed is still very vast. In this slide, you can see the red spots are the already existing natural proteins, This black spot is de novo designed protein, and this one is natural proteins that were redesigned. The gray area is the area of possibility of proteins to be designed, and with this much vast possibility, we can introduce solutions to many modern day challenges in terms of medicine, in terms of technology, and in terms of sustainability. With this, I would like to conclude its journey. Thank you.
Thank you, Mr. Astabhuja Mishra, for simply masterful presentation on Baker's research journey about the designing of plethora of new proteins and the vast impact of these proteins in different areas to benefit mankind, and also make us capable to go beyond the creature's creation by designing completely new different kinds of protein. So now again we can turn the question around and ask how the structure of protein may be determined by knowing the sequence of amino acids. We all know that how the AI has been making inroads and tremendous impact on our daily lives. Demis Hassabis and John Jumper make use of this ingenious tool to solve the protein folding puzzle. So, to understand how they contributed in solving this long-standing protein folding puzzle by applying some critical algorithms, we have Mr. Imran Ansari, who is working with Dr. R. Ravi Shankar. Over to Imran now.
So good afternoon, all of you. Thank you, Shilpa, for introducing me, and thank you, Basit and Astabhuja, for giving us a brief introduction about the introduction of artificial intelligence and deep learning in the field of structural biology, and obviously the Baker's journey in designing a protein. Today I'm here to discuss this invention that is called AlphaFold 2, and this has been developed by these two scientists. The first one is Demis Hassabis. He's a British artificial intelligence researcher, graduated from the Queen's College, Cambridge in 1997, and where he completed his computer science tripos, which enabled him to directly enter to PhD in the University College London. Then after that, after completing his PhD, he founded and is now the acting CEO of Google DeepMind. Recently, in the year 2021, he has also founded another company which is called Isomorphic Labs. That company is more into drug discovery pipeline and has collaboration with another companies like Eli Lilly and Novartis. The another Nobel laureate is John Michael Jumper, born in 1985, graduated from Vanderbilt University in 2007, did his MPhil from University of Cambridge in 210, completed his MSc in theoretical chemistry from University of Chicago in 2012, and then entered his PhD and from the same university. Although the initial meetings of Jumper and Hassabis are not publicly documented, but their shared academic background suggests some natural type of convergence, which resulted in the invention of this AlphaFold 2. So before going into the structural and the functional aspect of this AlphaFold 2, we should know about this prestigious competition, sort of Olympiad, occurred in every 2 years globally, that is called CASP, the Critical Assessment of Protein Structure Prediction. It has been started by John Moult in the year 1994 in the University of California. Their aim is to, they give the target proteins to the predictors around the globe, and the predictors predict the structures within a prescribed times like 3 to 4 weeks, and then they submit the predicted models, and these assessors compare these predicted models with the experimental structures of these structures. So there are various parameters like the root mean square deviation, the template modeling score, and the global distance test score, among many others. This is the graph since the year CASP commented in the year 1994. As you can see, till CASP 12, that is the year 2016, no predictors or the no teams were hardly reaching to an accuracy of more than 60%. But this whole landscape changed when the AlphaFold entered into this competition, and achieving an accuracy of 72% in the year 2018, and again achieving a never seen accuracy of almost 90% in predicting the structures. This leap is actually what draws the attention of the structural biologists and the bioinformaticians across the globe in looking into this AlphaFold, like how does it function and what are the parameters on which it relies. So this means that there has to be a developmental story of AlphaFold from reaching to 90% and starting from 60%. So in the year 2017, Hassabis and his teammates, they constructed a computer model based on the convolutional neural network. As the name suggests, this convolution is based on a mathematical operation. This means that the main focus is on the local regions of the query instead of scanning all the sequence at once. So with this type of neural network system, they entered in the CASP-13, that is in the year 2018, and secured first rank with an accuracy of almost 60%. After this, John Jumper, the computer scientist, joined in the year 2019 the DeepMind, and obviously he's a computer scientist, so he has that knowledge of advancing this technology. Jumper, Hassabis, and other team members, they abandoned this convolutional neural network and adopted the most advanced form of artificial neural network system, that is a transformer. So as the name suggests, it transforms the raw input file into a more redefined, more display type of output. So with this neural network, they entered the CASP competition again in the year 2020 and achieved this remarkable feat of almost 90% accuracy. So this draws the attention, and yes, these are some of the examples which the team members of the CASP they put as the targets. As you can see, AlphaFold 2 has achieved almost 99% accuracy in some of the targets, with the lowest being 90.5%. So in terms of achieving the atomic accuracy, AlphaFold 2 is doing very good. And now, obviously, we need to find how it works and what are the architectural, backbone of this AlphaFold 2. There are mainly two modules. One is EvoFormer and the structural module. They are the specificity of AlphaFold 2. The databases are already there, like PDB, UniProt, PFAM, Jackhammer. They all are the databases of protein, like the structural database, the family of the proteins, the sequence of the protein. They are already there in the database. So what does this EvoFormer and structure module do, and how does this database interpret when we put up a input amino acid sequence? We will see them individually. So talking about the database search and pre-processing, when we put up an input sequence in the database bar of AlphaFold, it is scanned in three ways. The first one is this genetic database search, in which the sequence is compared with the existing sequences, or like it may be co-evolved with other species also, and based on that, matrix is prepared, which is called the multiple sequence alignment matrix. And similarly, in the case of pairing, obviously when a protein needs to be folded, there may be some residues which may be closer to each other, and then there will be some residues which may be very far from each other. So in that respect, a matrix is prepared, and then there is the third type of scanning, which is called structural database search. Maybe the sequence which we put may be having already experimental structure in the database of the PDB. So based on that, a template and a sort of histogram is made, and combining all of this, the AlphaFold 2 got two types of representation. The one is MSA representation, and the another is pair representation. You can see that there is a gradient of color in both the matrices. The lighter color in MSA representation denotes that these two residues are very poorly likely to be co-evolved, and the darker residues, the darker boxes, means that the residues are very conserved in the course of evolution. Similarly, in the case of pair representation, the lighter color denotes that these two residues are very far in the scenario of folding, and likewise the darker color denotes that these two residues are very near in the scenario of folding. So these two representations are basically the substrate for AlphaFold2 neural network, and now they will enter the main structure, the main module of the AlphaFold2. So the neural network of the AlphaFold2 is named as EvoFormer, inspired from the name Transformer Neural Network. This EvoFormer neural network is composed of two separate towers. One is MSA representation tower, and the another is this pair representation tower. These two towers communicate with each other. They exchange information and updates each other several times to to get the results. There is a proper functioning of this EvoFormer. I'll put up. I have put up an animation in the next slide. We'll see that how this EvoFormer works. And after several cycles, we got the refined MSA representation and the refined pair representation from the EvoFormer as an output. So how does the MSA representation work? The network in the MSA representation prioritizes the row-wise analysis of the residue first, and then followed by the analysis in column-wise. Obviously, this is very important in context of the residues, like how they are closer to each other. And in the same way, the pair representation network does the job. It considers the residues as nodes, and the distance between two residues is calculated with the help of another residue, which is called as helper, the third residue K. And the distance of these, all the three residues, is triangulated in the form of a triangle. Like we know this theorem that the sum of the two sides must be equal or greater than the third side. So whichever residues satisfy these problems, they update the matrix. So how does these two towers communicate and work with each other? So before starting pair representation, the MSA sends the updated MSA to the pair representation, and based on that, pair representation sends back the information to the MSA representation for another cycle. So in this way, a total of 48 rounds occur in the EvoFormer before we get up the refined MSA representation and the refined pair representation. So now these refined representations of MSA and pairwise, they entered into the structure module. The structure module is built in such a way that it can perform three functions. One is the rotation, the translation, and physical and giving the physical and chemical properties to the residues. The first one is rotation. Obviously, when putting up all the amino acid residues in the 3D space, their orientation, their shape, their sizes needs to be fixed. So that happens in the rotation step. Then there is translation. So when the residues shape, orientation, and size is fixed, their location with respect to each other now need to be allocated. And after that, the bonding between them, the physical bond, the torsion angles, the bond angles will now be calculated, and all of this will lead to a proper 3D coordinate having the proper 3D structure of a protein. So this is the whole picture, like how AlphaFold2 works. 48 cycles of the EvoFormer, 8 cycles in the structure module, and total of 3 cycles they communicate with each other to give us a proper 3D coordinate to minimize the error and maximize the accuracy. This invention was published in the year 2021 in Nature, and this was the most highly cited paper in that year. Obviously, with this fabulous job, they collaborated with the European Bioinformatics Institute. You can see that in the protein database, the number of structures is around 200,000, while the number of the predicted structures in the AlphaFold database has been increased to almost, in fact, more than 1,000 times from the PDB database. So yes, it is doing a great job in terms of predicting the structure. Now, obviously, the most important aspect for us is how we can use it in our research work. So the first one is drug discovery. In the time of COVID, researchers in the US, and mainly these two companies, the Pfizer and Moderna, they predicted the structure of the 2P variant of a spike protein. This variant was highly immunogenic, so that it can be used to produce more amount of antibodies in that specific time. Now there is another application which is called effect of genetic variant. In this, the researchers predicted the structure of PINK2 mutated protein, which is a very important protein in Parkinson's disease. So now researchers are in the stage of screening drug candidates with this mutated protein. Then there is how we can model the protein-protein interaction. As we all know, this is the era of antimicrobial resistance. Bacteria are trying to evade the antibiotic response by forming novel complexes, novel proteinaceous complexes. So we can have look before going in vitro. We can form novel complexes of the protein and study them before doing the experiment in our wet lab. The another thing is engineering artificial proteins. So in University of Portsmouth, researcher used the Rosetta program of the Baker, and they designed such proteins which can basically eat plastic. So, and they are in the phase of screening some candidates which can do it more effectively. So there is application in sort of combating plastic pollution also. Now I have put up a video here also. All of us and any one of us can go to the system and access AlphaFold. Just 2 or 3 clicks job. All you need to know is your protein's name. The interesting part about this video is that the experimental structure of this protein is not yet known. Yet this AlphaFold 2 has predicted its structure nearly very in accurate way. Then there is another slide. It's somewhat anti to the AlphaFold 2. You can see the experimental structure of human insulin is very clear, while the AlphaFold 2 predicted it in a wrong way. Almost 30% of the residues are in high confidence, and rest the 70% are in low confidence. So yes, there is a lot of room for achieving accuracy in the case of AlphaFold 2. And also AlphaFold 2 can only predict the structure in its monomeric form, not in the oligomeric state. So I hope, and we should hope, that this problem may be addressed in the next versions of the AlphaFold, like AlphaFold 3 is on the way. So that's all from my side. Thank you.
Thank you, Mr. Imran Ansari, for presenting the typical algorithmic part into a very simplest way. Interestingly, this group of Demis Hassabis and John Jumper is active in all sorts of different areas, like creating games and weather forecast, and also creating Gemini, which we are using in our mobile phones. So in conclusion of the this session 1, we must say that in order to understand how protein works, we need to know actually what they look like, and this is what this year's Nobel laureates have done. So now I would like to end this session with a cynical joke that was made on the BBC radio after the announcement of the Nobel Prizes, that AI winning physics and chemistry. So to this, the professor who was talking answered that perhaps the AI had made the nominations as well and decided they would win it. So now we will have a short tea break of 15 minutes, and high tea has been arranged in the corridor in the left side, and after that we'll meet again for the second session, which will be moderated by Ms. Dipansi Saxena. Thank you.
हेलो, हाई टी का डिब्बा आप लोग को बाहर खा के आना है, अंदर नहीं खाना है। डिब्बा अंदर नहीं खाना है। ठीक।
हेलो 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 आ रही है? हेलो हेलो, आप लोग ऑडिटोरियम के अंदर ये सब ना खाइए, बाहर खा के आइए। हेलो
Welcome back, one and all present here. I am Dipanshi Saxena, your host for the second session of our ongoing 7th Nobel Symposium. In this session, we will dive deeper into the discovery of tiny gene regulators, that is microRNA. These tiny switches tune the expression of gene depending on the cellular needs. This year, Nobel Prize in Physiology or Medicine was jointly awarded to Dr. Victor Ambrose and Dr. Gary Ruvkun for the discovery of microRNA and its role in post-transcriptional gene regulation. The two Nobel laureates investigated mutant C. elegans nematode with developmental defects due to alteration at Lin4 and Lin14 genetic loci. Here are some clips and clicks of yesterday when they were awarded with the Nobel Prize.
Dear Professors Ambrose and Ruvkun, on behalf of the Nobel Assembly at the Karolinska Institutet, it is my great honor to convey to you our warmest congratulations. And I invite you now to step forward to receive the Nobel Prize from the hands of His Majesty the King.
Without giving any more spoilers, I would now like to invite on stage our first presenter, Ms. Neha Agrawal, who will take us through the captivating journey of our first Nobel laureate, Dr. Victor Ambrose. Neha is a CSIR-SRF working under the supervision of Dr. Prabhat Ranjan Mishra in Pharmaceutics Division. She has done her bachelor's and master's in biological sciences from Delhi and is currently working towards development of bone-specific nanotherapeutics for breast cancer bone metastasis. Over to you, Neha.
Thank you, Dipanshi, for such a warm introduction. Since we are back after a tea break, I would like to begin my talk with a question. Does this picture look familiar to all of you? Of course. The picture states the central dogma of molecular biology, according to which DNA is transcribed into messenger RNA. Messenger RNA is further translated into proteins, which carry out our cellular function. Now, our organs and tissues are made up of different kinds of cells, but all the cells contain identical genetic information stored in the form of DNA. But still, all cells carry out different functions as they express different kind of proteins. How is this possible? The answer lies in the precise gene regulation, which makes sure that specific kind of genes are expressed in specific kinds of cells. This gene regulation also needs to be fine-tuned so as to adapt to the changes in the cell as well as the environment. If this gene regulation goes awry, it can lead to diseases such as cancer, diabetes, and autoimmunity. Thereby, understanding the mechanisms of gene regulation has been a hot topic since few past decades. This year's Nobel laureate gave us a new mechanism of gene regulation which turned out to be very significant and important for humankind. Today, it's my honor and absolute pleasure to walk you down the journey of Victor Ambrose that led to a major breakthrough in the form of discovery of microRNA. Now, before we go to the life of Nobel laureate himself, I would like to take a minute to talk about Caenorhabditis elegans, which was the model behind his work. Sidney Brenner introduced C. elegans as the model organism 5 decades ago. It is named after the Latin word for elegant because of its wavy sinusoidal motion. Its salient features such as small size, rapid life cycle, body transparency, and ease of genetic manipulation have facilitated scientists to use it as a model for studying complex biological principles in a very easy manner. Now, this isn't the worm's first brush with international stardom, nor is it the first time that it has been thanked for award-winning work. In fact, it was the model behind the 2002 Nobel Prize in Physiology or Medicine won by Sidney Brenner, John Sulston, and Robert Horwitz. 2006 Nobel Prize in Physiology or Medicine won by Craig Mello and Andrew Fire. 2008 Nobel Prize in Chemistry won by Shimomura, Shalfie, and Seen. In fact, Victor Ambrose and Gary Ruvkin's 2024 Nobel Prize in Physiology or Medicine is the fourth award based on C. elegans research, cementing the worm's outsized role in scientific achievements. Victor Ambrose was born on 1st December 1953, Hanover, New Hampshire, USA. He completed his undergraduate in 1975, his doctorate in 1979, and his postdoctoral fellowship in 1983, all from Massachusetts Institute of Technology. Then, he joined as a faculty at Harvard University from 1984 to 1992 and at Dartmouth College from 1992 to 2008. In 2008, he joined the University of Massachusetts Medical School and currently holds the position of Silverman Professor of Natural Sciences in Molecular Medicine, endowed by his former Dartmouth student, Howard Scott Silverman. For his doctorate degree, Victor Ambrose joined the lab of David Baltimore, who is a co-recipient of 1975 Nobel Prize in Physiology or Medicine for describing the interactions between tumor viruses and the genetic material of the cell. In his lab, he studied the poliovirus genome structure and function. Then, for his postdoctoral fellowship, Victor Ambrose joined Robert Horwitz's lab, who again is a 2002 Nobel laureate in Physiology or Medicine for describing the genetic regulation in organ development and programmed cell death. It was in his lab when Victor Ambrose was first introduced to C. elegans as a model organism. Robert Horwitz had brought certain worm mutants with him back from Cambridge, out of which one of the strains contained mutation in LIN4 gene, which controlled the developmental stages in C. elegans. In his lab, he collaborated with another lab fellow, Gary Ruvkin, and they both started to study the heterochronic genes in C. elegans. They noticed that LIN4 loss of function mutants showed a phenotype in which there was absence of adult structures and also prevention of egg laying. Also, they noticed that LIN14 null function mutants showed a phenotype which was completely opposite to the LIN4 loss of function mutants. Victor Ambrose also noticed that LIN4 negatively inhibited the expression of LIN14 and LIN28. LIN14 and LIN28 inhibited the expression of LIN29, which ultimately controlled the developmental switch from larva to adult in C. elegans. He also noticed that multiple tissues expressed LIN4 and LIN14. So, after noticing that there was a gene regulatory cascade and tissue-specific expression, he came down to a conclusion that heterochronic genes exist in a hierarchy, in which some genes such as LIN4 exist at the top of the hierarchy and control the expression of downstream genes in a tissue-specific manner. In 1992, when he joined Dartmouth College as an associate professor and set up his own lab, he, along with his lab members Rosalind Lee and Rhonda Fenbaum, started to clone the LIN4 gene using restriction fragment length polymorphism and southern blotting, which was a painstaking task itself given the technology available at that time. After multiple open reading frame sequence predictions and clone resequencing, they started to suspect that maybe LIN4 is a non-coding gene. Further, they introduced frame shift mutations in the C. elegans sequence, which confirmed their sequence as LIN4 had a very short open reading frame. So further, they started to probe the LIN4 transcript using northern blot and RNase protection assay, which led them to the finding that LIN4 codes for two small transcripts, LIN4L, which is 61 nucleotide long, and LIN4S, which is 22 nucleotide long. Then as a next step, they started to compare the sequences of LIN4 and LIN14. And they found that the non-coding LIN4 RNA had sequences complementary to the 3' untranslated regions of LIN14. Now, 3' untranslated region is generally that part of the mRNA which is not translated into protein, and up till 1990s was only known to confer stability to the mRNA. The possibility that it could contain any kind of complementary sequences for binding of short RNA, or that it could play any kind of role in gene regulation, was not known up till now. So, after independently deducing the sequences of LIN4 in Victor Ambrose's lab and LIN14 in Gary Ruvkin's lab, on the evening of June 11, 1992, they exchanged the sequences and came down to a conclusion that yes, the non-coding RNA of Lin4 has complementary binding to 3' untranslated regions of Lin14. This was a moment when years of work came together as a clear model, a classic eureka moment. They both published their findings in 2 independent research articles in the same issue of the Cell back to back, in which the Victor Ambrose publication marked the discovery of the first microRNA Lin4, and Gary Ruvkin's publication was more about the mechanism by which Lin4 inhibited the expression of Lin14 in a post-transcriptional manner. This will be described in more detail by the next presenter, Pranoy. Now, the findings showed a collaborative effort between the 2 labs, each discovery being integral for the relevance of the other. The discovery unveiled a new mechanism of gene regulation active in a post-transcriptional manner mediated by microRNAs playing important role in organ development and tissue function. After this primary discovery, many scientists around the world they started their work to find similar microRNAs in other members of the animal kingdom. But parallel to the cloning of other microRNAs, some scientists they devoted their research to understand the biogenesis and mechanism of functions of microRNAs. Now, there may be many strategies for transcription of microRNAs. The canonical microRNA is transcribed by RNA polymerase II to form a primary microRNA, which is a hairpin loop structure. This primary microRNA serves as a substrate for binding of a microprocessor containing Drosha endonuclease that cleaves the strands to form pre-microRNA. Now, this was also first studied in the Victor Ambrose lab itself. Pre-microRNA is then transported from nucleus to cytoplasm by exportin 5 and RAN-GTP. Once this pre-microRNA reaches the cytoplasm, it serves as a substrate for binding of dicer endonuclease, which forms microRNA duplex. Now, out of this microRNA duplex, the effective strand is loaded onto the Argonaute protein containing RISC complex, and the passenger strand is degraded. Now, once the mature microRNA binds to its target mRNA in a sequence-specific manner, it can shut down the protein expression either by mRNA degradation or translation inhibition. Intriguingly, one microRNA can regulate the expression of multiple genes, and multiple genes can be regulated by one microRNA, thereby fine-tuning the genetic regulation pathway. Victor Ambrose lab at UMass Gen School is devoted to studying of microRNAs and the genetic control of animal development. His whole work is based on using C. elegans as the model organism and thereby understanding the contributions of microRNA in genetic regulation. Victor Ambrose is a passionate researcher who has contributed a large portion of his life to curiosity-driven research characterized by creativity, perseverance, open-mindedness, and data sharing, which have led to some fundamental findings important for humankind. Starting from the discovery of Lin4, the first microRNA in 1993, to present day, he has made some major contributions to the understanding of microRNAs in animal development. He has been awarded with various recognitions for his scientific achievements. He was elected to the National Academy of Science in 2007. He has received the Gardner Award, the Dixon Prize, and the Breakthrough Prize in Life Sciences. Also, he is a co-recipient of Lasker Award, Wolf Prize, and the March of Dimes Prize in Developmental Biology alongside Gary Ruvkin. And of course, the cherry on the cake being the 2024 Nobel Prize in Physiology or Medicine, which he again won alongside Gary Ruvkin. The collaboration, which started in the very initial days of their career as fellow postdocs in the same lab under the supervision of Robert Horwitz, definitely came a long way ahead and led to the winning of the most prestigious award in the scientific community, the Nobel Prize. This is the picture of Victor Ambrose and Rosalind Lee after the Nobel Prize announcement on October 7, 2024. Rosalind Lee is not only his partner in life, but is also his partner in science and is a co-author of the iconic historic 1993 paper marking the discovery of Lin4. And this is a short clip which shows the celebration at UMass Gen School after the Nobel Prize announcement.
In every way, all all of us. We feel like we won the Nobel Prize, you know. One piece of credit that you have neglected, and you may not think about what that is, but here's a little reminder. This place has never won a Nobel Prize for anything but war and biology.
We were joking the other day when I first asked Candy out in 1972, did I know that this would be the key, you know, to the Nobel Prize, right? You know, so.
It's just incredible what we've been able to do together, you know, and there's so much more to do, and you know, we're not done yet, and the worm's not done yet. Eight gold medals, 4 Nobel Prizes, and it's still got a lot of secrets to uncover.
Thank you.
Thank you, Neha. This was really an inspiring journey of observations and conclusions. Now, I would like to invite my second presenter, Mr. Pranay Topo, who is UGC SRF working under the supervision of Dr. Amir Nazir. He has done his bachelor's and master's in zoology from University of North Bengal, Siliguri, and is currently working towards mitochondrial quality control and its role in neurodegenerative diseases, employing C. elegans as a model. He will take us through the scientific journey of our second Nobel laureate, Dr. Gary Ruvkun. Over to you, Pranay.
First of all, good afternoon, everyone. Thank you, Dipanshi, for the brief introduction. Today, I will walk you through the journey of Professor Victor Ruvkun towards unveiling the extensive role of microRNA in eukaryotic gene regulation. So Professor Ruvkun was born on March 26th in the year 1952 in Berkeley, California, US. He did his bachelor's majoring biophysics from University of California, Berkeley in the year 1973, followed by his PhD from Harvard University in the year 1982. After completion of his doctoral work, Professor Ruvkun joined MIT, the lab of Dr. Robert Horvitz, for his postdoctoral research. Currently, he holds the position of professor in Department of Molecular Biology in Harvard Medical School. So for his doctoral studies during its time in Harvard University under the supervision of Dr. Frederick M. Oswald, his PhD supervisor, Professor Ruvkun was working on the bacterial nitrogen fixation gene. It is in MIT during his postdoctoral research, Professor Ruvkun comes under the first contact of worm biology, and he was working under another Nobel Prize winner, Dr. Robert Horvitz. It was there, under his supervision, Professor Ruvkun published few of the seminal research articles describing different aspects of microRNA towards regulating developmental timing in C. elegans. So currently in Harvard Medical School, Professor Ruvkun's lab currently works on 3 research trajectories, namely the microRNA genes and other small RNAs, control of longevity and immune surveillance, and the detection of life on other planets. After the first discovery of first microRNA, that is LIN4 by Professor Victor Ambrose, Gary Ruvkun contributed immensely towards understanding its mode of action. That is, how LIN4 microRNA targets the LIN14 mRNA and ultimately controls its pattern of expression. Not only that, Professor Ruvkun also discovered the second known microRNA, that is LET7. And both of our present laureate contributed immensely to understand the mechanism of action of microRNA, how they target different mRNA to regulate the developmental timing in C. elegans. In this area, Professor Ruvkun played a tremendous role in showing us how lin-4 microRNA targets the lin-14 mRNA and binds to the 3' untranslated region and ultimately forms a conserved loop that is very much conserved throughout the different species, and how it leads to the degradation through a conserved mechanism. These two research articles are considered as major breakthrough from Professor Rafkun's work describing the different aspects of microRNA and how it regulates the heterochronic gene expression, that is lin-14, and mediates the temporal pattern formation in C. elegans. Not only that, he also discovered the second microRNA, that is 21 nucleotide long LET7, which also contributes in regulating the development of C. elegans. So Professor Rafkun showed us that by targeting lin-14 mRNA, the lin-4 microRNA forms a temporal level of expression of lin-14 protein. In this slide, in the leftmost panel, that is figure A, it is about the immunoblot analysis of lin-14 protein, where you can see the expression of lin-14 protein gradient. The proteins that were used here were harvested from age synchronized worms, and the strains that were used here are the wild type, the 2 heterochronic mutants, those are lin-14 gain of function mutants, of course, and the lin-4 mutant. In the second figure panel, that is figure panel B, this is the transcript analysis, the RNA transcript analysis of lin-14 gradient. And you can see that it does not follow the temporal gradient as it is shown in the protein level expression, which tells us a story that lin-14 instead is maintained at a post-transcriptional fashion. In the rightmost figure panel, that is the expression of LAC-Z reporter under the control of collagen 10 ANC54 transgene that is specifically expressed in the muscle wall of C. elegans. Here in the first figure panel, that is A1 and A2, we can see that the transgene is expressed in both the larval stages. But in case of B1 and B2, it is only expressed in case of early larval stages, which tells us that lin-4 plays a significant role in controlling the pattern expression of lin-14 protein at the early larval stages of the worm. In the bottommost figure panel, that is C1 and C2, here we can see that the transgenes is being expressed in both the larval stages. This suggests that here there is a disruption of lin-14 expression, and it is from lin-14 mutant. So Professor Rafkun conducted the previous sets of experiment, but this time he uses another species of worm, that is C. brighesi, and he showed us that it follows the similar trans-species expression of lin-14 temporal gradient. A paper which came on December 3rd, 1993, which considered as one of the breakthrough in the microRNA research, tells us a story about how lin-4 microRNA controls the expression of lin-14 mRNA. Here, Professor Rafkun showed that how lin-4 microRNA binds in the 7 different regions of its target lin-14 mRNA, of course at the 3' untranslated region. Here, the first 3 RNA duplexes of lin-4 and lin-14 mRNA that is shown by the blue color boxes, you can see forms at the very core region of the RNA duplex. But the other 4 sites, which are shown here in the yellow color boxes of lin-14 mRNA, base pairs with the lin-4 RNAs by bulging out one of the conserved C residues of the lin-14 mRNA. And after formation of this RNA duplex, that is of lin-4 and lin-14 mRNA, their degradation takes place. Back then, the mechanism of RNAi or RNA interference was not known to us, and that is why Professor Rafkun has coined the term accessory proteins and some sort of catalytic activity of this RNA duplex. But later on, through the extensive research in this field, today we know that there are some conserved mechanism of RNA interference exerted by these proteins, which played a tremendous important role in controlling the gene expression. From extensive work in this field, Professor Rafkan also showed that how heterochronic genes or the different other microRNA controls the level of development at the different stages of the worm. In the leftmost figure panel, he showed us how LIN4 and LIN28 act antagonistically to ultimately regulate the expression of LIN14. Here, the work that is carried out or led by Professor Rafkan showed us that LIN28 acts antagonistically to LIN4, and it is very much important for smoothly transition of worm from L1 stage to the later larval stages. And he also found out that the LIN28 also controls the expression of LIN14 in case of LIN4 mutant and other two heterochronic LIN14 gain of function mutant. In the rightmost figure panel, it is about the heterochronic regulation of this microRNA to the other targeted mRNA. Here in this first column, you can see that LIN4 controls the expression of LIN14 and LIN28. And this is very much important for worm to smoothly transition from L1 larval stage to the other larval stage. In the other larval stage, that is from L2 to L3 transition, the regulation of other microRNA, that is LIN41, takes place through another conserved microRNA, that is LET7. So here, this LIN41 silencing is very much important to release its expression of LIN29 microRNA, which is another important microRNA which helps the worms to smoothly transition from L2 to L4 larval stages. So, besides working in LIN4 biology, Professor Rafkan also discovered the second known microRNA, the 21 nucleotide long LET7, and he showed that unlike LIN4, LET7 microRNA is conserved through many other multicellular life forms, starting from porifera up to us, that is human Homo sapiens. So here you can see that LET7 has a sequence conservation, and it codes for around 21 nucleotide length chain that significantly bind to its targeted mRNA and forms a conserved stem loop structure. It first discovered in C. elegans, but later on Gary Rafkan showed that it is conserved, the mechanism of its action is conserved in other species such as Drosophila melanogaster and Homo sapiens. Here one interesting fact is that the work led by Professor Rafkan showed us how two microRNA, that is LIN4 and LET7, controls the pattern of development at different life stages of the worm. Here, from the work of both our Nobel laureates, we now know that LIN4 controls the level of expression of LIN14 gene at the early stage of development, that is from L1, L2, etc. But the LET7 microRNA, which is the second discovered microRNA, controls the development at the later larval stages and specifically allows the transition smoothly from L4 to the adult stage. So after the discovery of LIN4 and LET7 microRNA, today we now know that microRNA comes under a class of non-coding regulatory RNA, which plays a various important role in biology that not only restricts in post-transcriptional gene regulation, but other aspects as well. So from Professor Rafkan's work, today we know that not only worms, but other species such as Drosophila and humans do contain the microRNA loci, and they are around 163 in number in case of Drosophila, 575 in case of humans. And this all made possible through the research contributions made by the researcher and scientists all over the world. And it is not the work of a single person. So here I would like to tell a little story about the perseverance and the belief. So the two Nobel laureates here, that is Dr. Craig Mello and Andrew Fire, who won the Nobel Prize in Physiology or Medicine in 2006. Actually, Professor Craig Mello was the PhD student of Victor Ambros, and they discovered the mechanism of RNA interference back in 1998. And by the year 2006, they won the Nobel Prize in Physiology or Medicine. Back then, it was the quickest Nobel Prize ever given to any science category. It shows that how tricky and unpredictable the field of science is. After the announcement of the Nobel Prize in Physiology or Medicine for RNAi discovery, many of the scientific community thought that the story or the discovery of microRNA is already being covered in their award. But here we now know that after winning the Nobel Prize in Physiology or Medicine by Professor Rafkun and Professor Ambrose, that science, the path of science does not always follow the traditional hierarchy. So it also tells us about the role of perseverance and secrets, which is ultimate for all triumphs. So after the discovery of microRNA back in 1993 up to present year, it has tremendous amount of application in many aspects of modern biology that not only restrains in understanding the gene regulation, but also in the various areas of modern biology. So from here on, my fellow presenter Dipanshi Saxena will be talking about the different areas of application of microRNA in drug discovery. Currently, Dipanshi Saxena was as UGC SRA 4th year student under the supervision of Siddharth Chopra, and he is currently working in the area of identification and detailed biological characterization of antibacterial compounds against human pathogenic bacteria. So by this, I would like to invite my fellow colleague Dipanshi, who will take forward the talk. Thank you, everyone.
Thank you, Pranoy. You beautifully explained how LIN4 regulates LIN14 during the developmental stages of C. elegans. Now, we will look into the application aspects of microRNA in drug discovery. With the ability of microRNA to regulate gene regulation, gene expression, these are involved in a variety of biological processes, including cell cycle, cell differentiation, proliferation, apoptosis, energy metabolism, etc. This involvement of microRNA in almost all the cellular process have led to the exploration of its role as biomarkers in target identification or as target for therapeutic interventions. Historically, there was a belief that RNA molecule could not be a suitable biomarker due to the presence of endogenous nucleases within blood samples. However, the theory was rapidly dismissed with the discovery of microRNAs in the fixed tissues, where they were consistently present and were stable in the serum samples. This led to the discovery of exploration of role of microRNAs as biomarkers in several diseases including cancer, cardiovascular disease, autoimmune disorder, neurological disorders, and also the infectious diseases. Although several microRNAs shows redundancy in their expression, but there are certain specific microRNAs which are either overexpressed or repressed in particular disease condition only. These circulating microRNAs are explored as non-invasive biomarkers for quick and easy identification of the disease. The advantages of using microRNA as biomarkers starts with their quick isolation from the tissue sample or the blood samples. Secondly, they are comparatively more cost-effective and less time-consuming when compared to the protein antibodies. And lastly, they can be used as multi-marker model system for accurate diagnosis of a disease. Now, the pathway of taking mRNA into the drug discovery process starts with identification of the signature microRNA followed by the mRNA profiling. This is followed by validation of the signature mRNA by conducting loss or gain of function studies in both in vivo and in vitro conditions. Thereafter, the microRNA molecule is taken for its pharmacological analysis, where the first and the most important step is delivering the mRNA molecule into the in vivo system. This in vivo delivery of microRNA is aided by associating the microRNA molecules with viral particles or by encapsulating it into lipid-based systems, polymers, or as inorganic nanoparticles. After the successful delivery of microRNA into the in vivo system, the molecule is taken for its toxicity and efficacy studies. Thereafter, the studies are taken into the clinical trials starting from phase 1 to phase 3, followed by its approval in therapeutics. Several studies have revealed that targeting microRNA have proven to be an effective way of disease suppression. Here are some examples where the targeting specific microRNA associated with particular disease have led the study to the clinical trials. For example, MIR-122 is specifically targeted in hepatitis C virus infections. MIR-122 is a highly abundant liver-expressed enzyme which binds to the 5' untranslated region of the hepatitis C viral genome. This binding mask protects the viral RNA molecule from host nucleases mediated degradation. In this case, a experimental drug miravirsin was introduced into the by Centeris Biopharma for the treatment of hepatitis C virus. The drug is an oligonucleotide of 15 base pairs and which forms an heteroduplex which is very stable with MIR-122. This removes the host-mediated protection to the viral genome. The drug has established its safety in phase 1 clinical trials and efficacy in phase 2 clinical trials. Not only in abroad but also in India, scientists all around are working towards exploring the role of microRNA as biomarkers or in diseases, particularly cancer. Some of these institutes include IISc Bangalore, IIT Madras, Cancer Institute, AIIMS Delhi, AIIMS Bhopal, etc. Also, scientists at our institute are actively involved in exploring the role of microRNAs in either the developmental processes or diseases such as cancer, osteoporosis, etc. Although microRNA-based therapies have proven to be an effective way of disease suppression, but there are certain limitations of the therapy too. Firstly, the mRNA-based therapies need an in-depth optimization of parameters such as age, gender, disease, reproducibility, etc. because these parameters changes with individual. Secondly, the delivery and stability of microRNA in the in vivo system is a matter of concern. Lastly, the robustness, the redundancy of microRNA makes it difficult to study the role of single functional microRNA in particular disease. However, the robustness of the system explains how the system is not easily manipulated by, for example, viruses. To conclude the session, as we now know that microRNAs are present in almost all the living organisms present on Earth, the expression and expansion of microRNA genes are intimately tied to the evolution of more complex organisms. The discovery of microRNA have given a new dimension to the gene regulation and explains how an organism develops, function, and how a disease progresses and how it suppresses with the therapeutic interventions. Thank you. Now, I would like to invite Director Ma'am on stage to kindly felicitate our participants of this session. Starting from the chemistry session, I would now first like to invite Mr. Abdul Basit Khan. Mr. Ashtbhuja Mishra. Mr. Imran Ansari. The moderator, Ms. Shilpa. Moving on to the biological session, Mr. Pranay Topo. Miss Neha Agarwal.
The moderator of biology session, Ms. Dipanshi Saxena. Now we would like to invite our mentors also to join us. Dr. Imran Ansari, Imran Siddiqui, sorry.
Dr. Kinshuk Raj Srivastav. Dr. J.V. Pratap. And Dr. Amir Nazir.